I'm Lorenzo. I'm a postdoc in the Albert Lab. And today I would like to tell you more about the research that I have been doing in the, here about using population-based modeling for identifying the functional signature of genome organization. So Taylor already gave a very nice introduction of the general notion here. We're all here to investigate chromatin organization. We know the chromatin organization is multi-scale and is really functional. So this very specific organization is really necessary to be able to code the different biological functions, such as the regulation or the transcription and the regulation. And what we do in the lab, we adopt a computational approach to try to answer the following questions. So first of all, how can we generate accurate models for genome structure? And also later, how can we use those models to get a peek into the structure function relationship? So how a particular structural motif impacts and codes for biological function. And I will try to to give a brief overview of both strategies here. So I'll start with the strategy that we use to model the genome, and we generate population of structures from data. So there are two key concepts here, which are the data itself and the population of structures. So as for the data, the rationale is actually very simple. Over the years, a lot of different experimental assays and technologies have been developed to probe essentially derivative features of genome organization, either distances or shapes or spatial proximities to nuclear landmarks. And so if we can do this, how about we reverse this notion? So how about we go from this collection of independent experimental assays to generate back the actual three-dimensional organization of the genome? And I like to think about this process as a fancy genome reverse orthographic, where basically you have the actual three-dimensional object, that is the 3D genome per se, and you have all these projections in terms of feature spaces, and you're trying to combine that information to recreate the actual object. Sometimes, as you can see from this schematic over here, using just one projection, so for instance, just one set of data, is not quite sufficient to nail down the ambiguity. You still have a lot of ambiguity in how maybe a structure or structures may look like, and so you need to combine that information with other data types. This way, you're really funneling down towards the only realistic structures that are really compatible with everything else. So so this is the first thing. So we're using data. So we're trying to generate structures that recapitulate a variety of data that we use. And also, the key notion is that of population of structures. So in our case, we try to capture the heterogeneity of the genome by describing that with a collection of structures, of unique single cell structures. So when you pull together all the structures, you get a population, and that is what we use to represent the genome. And let me just briefly sketch how we implement our data-driven process here. And I'll do that using the Hi-C data because this is easier to show actually visually. So we have an experimental contact map that we would like to use as a generator for our population of structures. And clearly, we would like the population of structures to predict a very similar contact map. So how do we actually do that? So basically, we allocate the contacts. We distribute the contacts across the different structures in the population in such a way that, you can see these for these 3 examples, in such a way that each structure is possibly unique per se and distinct from all the others. But at the end of the day, when you pull the structures together, you do get the experimental, you do get the input contact map. And this is an iterative procedure, of course. To get more technical about this, what we do to solve the problem is we maximize the probability here that the structures express the input data. So this, as you can see, this is a joint optimization. So we optimize all the 3D coordinates of all the loci in all the structures, based on the experimental data that we have as an input. And that could be, like in this case, just the high C, or we can combine this with high C and MID and more. That's something that we have in place. And there's another element here in this optimization problem, which you probably noticed. This D star is another variable which we jointly optimize with the coordinates. And that is those are the data latent variables relating to the data. And this quantity contains information that's missing from such ensemble data. So for instance, when we assign the contacts, when we distribute the contacts across the structures, we go from probabilities to contacts. And so for instance, we need to decide which structures in the population express those contacts. And this information, which is missing here, is encoded in the D star. And that's why we have the D star here. So now, this data-driven deconvolution of data into a population of structures is now up and running, and it's available here on the lab GitHub. And we just recently published a method paper here where we really investigate the different benefits of having high seed, MID, fish, and sprite data simultaneously as inputs to generate a population of structures. So now the other thing that I would like to discuss is, so we have these models with these structures, and of course, as a sanity check, those structures recapitulate the input data, right? But that's not just good enough. The rationale here is you use more data together to really remove the ambiguity, and so hopefully you're getting such realistic structures that they also reproduce by default, by construction, other quantities that were not used in the modeling process. And so here, I would like to show some predictivity results from our high-C-based GM models that are about to appear in Nature Structure and Molecular Biology. I mean, specifically for this type of cell, for the GM cell, we saw that using high-C data is already a very good baseline, and including other data types only provides incremental corrections, incremental improvements. So I'm just showing results for the high-C data. So, and they are good already. So we have the structures, and we can predict very nicely distances to the nuclear landmarks. So on the x-axis, I'm showing the predictions from the models. On the y-axis, there are the experimental results, the signals. And so here we have nice correlation with the GP6 experiments in terms of radial positioning predictions. And we can also predict the location with respect to the speckles and to the laminas, also with very high accuracy. Moreover, we compared our predictions with results from the recently published DNA Murfish experiments paper. So here again, we are predicting very nicely the association frequency of the loci with the lamina, with the speckle, with the nucleoli, and even more quantities here. Something interesting is, besides very good correlation on a genome-wide level, we also saw that the structures, the chromosomal structures here for chromosome 2 that we get out of our simulation pipeline, they show the same structural variability that you can appreciate in experiment. Here I'm showing the comparison between the distance matrices for chromosome 2 from the experiment, so from DNA merfish, with the model. And the correlations are pretty nice, and the structural patterns are also pretty nice and variable. So the level of heterogeneity of our population of structures is somehow comparable with what people see in experiments. So this is for the modeling. So how we do the modeling of the structures, and to convince you that the structures are predictive. Now, now I would like to discuss. Now that we have this platform and the structures in place, we started looking into how can we use the structures to really characterize the functional signature of the structural features. And so, a notion that's crucial to us is what we call the microenvironment. So we have our simulated structures, and we can think about looking at them like under a microscope. So because we have all the coordinates of all the loci in all the single cells, we have access to a very wide amount of different structural features that sometimes experiments cannot probe at this level of coverage. So for instance, even though we don't use information about nuclear bodies, we designed a procedure to predict the number and positioning of the speckles of the nucleoli. And of course, we can predict for each locus its association with all these nuclear bodies and its positioning, its radial positioning inside the single cell. So this is for the global genome organization, but we can also access local features of the chromatin fiber, such as the local compaction, for instance. So for each of the loci, we can basically compute all these different structural features and more. And I'm showing here the signals for those structural features for all the loci in chromosome 1. Something that's critical here is because we have a population of structures, not only can we predict the average behavior. So for instance, we can predict the average radial position of a given chromosome locus, but also you can study its variance across the population, so its cell-to-cell variability. And this is true for many of these quantities. So we have the average trace, but also its variability. And the variability, I'm going to show you later, we propose is really connected to is a strong indicator for functional behavior. So now we can define all these quantities for each locus. So we end up with a feature vector for each of the chromatin loci. And in this sense, this feature vector spans a lower dimensional space, which is much lower dimensional than the three-dimensional space, than the high Cartesian space for all the coordinates for all the structures. And this space we call the nuclear microenvironment. And it is a mixture, again, of global organizational features, but also local organization features for the chromatin. And now I would like to convince you that some of the features in this microenvironment can really be helpful. This is a result from from a paper which is about to appear here. We computed for all the single cells in the populations, and for all the loci, we computed the cell-to-cell variability of the radial position, but also the mean speckle distance. This was done for the GM structure, and so we have a joint distribution of these two features across the population for all the loci, and this is what I'm showing here. And interestingly enough, you see that these two regions over here, so the red region and the blue region, which represent respectively loci with high speckle association frequency and loci with high lamina association frequency. So those loci, they display very, very low variability, which indicates their positioning within the nucleus is pretty homogeneous. And so we propose in the paper that those regions, like the nuclear speckles and the lamina, they provide structural anchor points around which the organization of the chromatin revolves, basically. And there is so much more about speckles that we can say from computing the microenvironment of the loci. And I totally suggest that you take a look at this paper, which is about to come out. We see already that the cell-to-cell variability of a given quantity can tell you something about functional organization of the chromatin. So, and now we can say even something more. Again, this is for the GM cell line from the same paper. I'm showing again the joint distribution of the loci in the cell-to-cell variability of the radial position versus radial position space for the A compartment and the B compartment, the classic compartments. And the loci are colored by subcompartment assignment. So based on the cell-to-cell variability of those loci, we can define 4 chromatin classes, A and B, with either high variability or low variability. And we can study the enrichment of those chromatin classes in terms of a number of features. So this is the enrichment plot, for instance, for those 4 classes in terms of subcompartment assignment, and there is clearly a pattern there, but also in terms of replication phases, and also in terms of lamina associated domains, for instance. And I'm not going into the details here. Those are discussed very nicely, again, in the paper, but it's clear that there is a strong functional signature here, and the enrichments are very different across the different chromatin classes. So this seems to indicate that structural variability can be a strong indicator for chromatin function. Something else very interesting that we can say, this has not been published yet, but we can also correlate changes in the nuclear microenvironment, so in those structural features, with changes in gene expression. So again, we can compute jointly a number of quantities in each single cell. And here specifically, we are focusing on the distance to the lamina and the distance to the speckle for a given gene. And we focused on 3 different genes that are involved in skin development, and we studied the microenvironment of those genes in both H1 and HFF cell line population models. So these 3 genes, first of all, they have different expression levels, as you can see here, in H1 and HFF. Here is downregulated in HFF, upregulated in HFF, and they have a very similar gene expression pattern here. So for those genes, we can compute indeed the nuclear microenvironment and study, again, the enrichment in both cell lines for all of these structural features. And you see that there's a lot of variability, both for the same genome, gene across the 2 cell lines, but also for the same cell line across the different genes. So there is major conformational rearrangement as the cell differentiates from H1 to HFF. And in particular, we're looking, as I mentioned before, at how the distribution looks like across the population in the distance to speckle versus distance to lamina space. You see that here, the 2 distributions, they are well separated in the space, whereas here, where the gene is upregulated in HFF, they tend to come closer, and there's also this very interesting, double peak distribution. And then eventually for the ACTB gene, when the gene expression is pretty comparable, two distributions overlap with one another. So this really showcases that using the microenvironment features can really be helpful to characterize characterize functional implications here. So I hope that I was able to convince you that we can really use that we can really use our models to say something relevant about the functional properties of the chromatin, and of course we're exploring this more. But now I would like to look back shortly, because while we're addressing those issues with structure-function relationship, in the same times we are aware that novel data types are being produced, especially single-cell imaging data types, and so we are trying also to work towards expanding the models to include more data types so that we can make the models more predictive. And so one thing that we're trying to do now is to make shapes more realistic. Like these are the models we've used so far, but we know that this is really not how things look like. And so we really would like to include information about the laminas and the nuclear bodies also in the models to make them more realistic. So ideally, this is something that we're doing now. So we are relaxing the chromatin together with high-C data, for instance, in very specific single-cell structural environments, let's call them that. And this is very interesting also because now we can append new structural features to this microenvironment, because before we didn't have features that described the laminas and the nucleoli, and this is something that now we'll probably do. So we can study also the variability of the microenvironment as a function of those new features. And finally, as we're talking about single-cell imaging integration, something that we can do now is to use single-cell chromatin tracing data to really produce, in combination with higher resolution data, to produce higher resolution single-cell models, as you can see here, starting from the DNA fish data. So really, we can use higher resolution data to trace chromatin between. And this is something that's in place, and we're right now the platform is in place, and we're looking for for collaborations, people who have this type of data and are interested in collaborating to us and make the most of this data. So over time. So let me just wrap this up. So I discussed the integrated genome modeling pipeline, which is a data-driven and multi-model platform to simulate a population of single-cell whole genome structures. I hope I convinced you that we can use the structures to define and characterize the microenvironment of genes, and that the microenvironment really allows to identify specialized chromatins and characterize functional properties. And finally, we are moving towards a revamped version of the IGM pipeline, and we're including single-cell imaging data. So I would like to thank the lab, especially Frank for his support, and of course the funding, the 4DM, and thank you for giving me the opportunity of speaking here, and I'll be happy to take questions.